Okay, welcome back. This next concept, concept 3, deals with prokaryotes. So prokaryotes are just like cells, just to give a very quick definition. So a prokaryote is it's like a type of cell. It's no organelles. That's very important. No nucleus. Those are very important characteristics. And I think it's all, at this level, all unicellular. These were probably from the first cells. This is the most, it's just very basic life, but also pretty highly evolved life in its own way, and we'll get into that. So the three things that I'm going to talk today with you about are like the early prokaryotes, and classification. So there are some techniques for classification. Then we'll talk about structure and composition. And then finally, we will talk about function and abundance. So let's first talk about like prokaryotes and the like different types that we have and how they sort of evolved. So let's talk about one. Prokaryotes and classification. All right. So early prokaryotes, right, were the first cells. So let's talk about cells. They're cells. So cell theory says all organisms are from cells. Cell is the basic unit of life. And all cells come from pre-existing cells. And this sort of there's like a ancestral cell. That's why it connects to that. So there was like one cell which sort of started all of this, that were all common ancestors too. And we can see in the record that cells have been along for a very long time, like they're very old in general. I mean, we have stromatolites, so these like fossilizations of bacteria in rock, and they're like about 3.5 billion years old. So very, very, very old. Very, very old. And during that time, we have the stromatolites living in a different environment. So this brings to a point, which is that Earth has changed significantly since old times. Old times, I say like someone was there, but since 3.5 billion years ago. So we've seen atmospheric changes. And the most notable change was increase of O2 at some point, and the introduction of free oxygen into the atmosphere, which basically, there was this big event. The theory is that there was a big event because photosynthetic bacteria used water to create glucose, to photosynthesize, and this bacteria, it's called cyanobacteria. It's hypothesized that, there's a theory that cyanobacteria change the composition through photosynthesis. So it leads to that, the cyanobacteria, and that led to atmospheric changes eventually. So it's very that reaction, that respiration photosynthetic reaction is very important for that. Was very important because it changed the composition of the Earth, of the Earth's atmosphere. If you took carbon dioxide in the environment and water in the ocean, C6H12O6. So too, if you watch the other videos, glucose, that's a sugar, it's food. So, we've had, cyanobacteria, it's been theorized that it's changed, and that relates back to how there are like old ancestor cells, and that there are fossil records of cells being quite old. This cyanobacteria was a early prokaryote. Now, there's some other important factors we have to consider, which is prokaryotes have certain functions. They do certain things. So we have just in general, that cellular structure correlates to cellular function. And that's just something to remember. Each cell is basically a functional entity. This is just important to remember in general when we're talking about prokaryotes, so that they have different structures. But in general, they share quite a few units are similar, and then the methods of DNA replication are all very similar. We share genes with the cyanobacteria, or E. coli shares genes with cyanobacteria. We all share the same genes. This is very important because it really reinforces this point that all cells come from pre-existing cells. Right. So another very important thing. Prokaryotes are very small, just very small. Prokaryotes are very small, especially when we compare them to eukaryotes, because eukaryotes are quite a lot more complex. Okay, so let's talk about classification now. We have three, so in the whole like massive, like coming from the first cell, we have like diversification, and two of the three like super kingdoms, the domains, are prokaryotic. So we have bacteria and we have archaea. Now these aren't as closely related as you'd think. They're definitely not the same. Archaea is more closely related to the third kingdom, which is eukarya, right? So more closely related than is to bacteria. There's a split off later. So let's look at like the differences between bacteria and archaea. So bacteria, archaea, and in the middle we'll do some similarities. So they look similar. Of course, looking is very different from being. Structurally very similar. But if we look at like when they were formed, then we see that closer to eukarya in genetic info, and this is like fairly different. Genetic information to eukarya and archaea. So if we look at like other differences, we look at like DNA mechanisms to replicate DNA. Archaea's mechanisms are much more similar to eukarya in general. Like there are multiple basically there are multiple DNA polymerases. So DNA polymerases, they like add bases when DNA is being replicated, and in this one there's one DNA polymerase, right? Does that make sense? I hope so. So if we look at like for example, the like bacteria, for example, has a peptidoglycan wall, like a cell wall. And archaea does not. Right, so one does and doesn't have a cell wall. They have some other type of cell wall. So they use I'm reading here pseudopeptidoglycan and other polysaccharides. If you look at the membrane, is also one of the interesting differences. The membrane here is a bilayer phospholipid, right? And in this one, it is most of the time, well, it's bilayer sometimes, but most of the time it is monolayer. They can have monolayer phospholipid. Membrane. So the membranes are different. Cool. So they look similar, but they've got differences. And realistically, and to be honest, archaea is just closer to eukarya than prokarya. Some of the things, these ones, they can produce resting spores. So this allows the molecule to like face bad conditions and to regenerate, like turn into a small bit of itself and then eventually be regenerated. And these ones have not been known to produce these resting spores. But a lot of archaea is seen to be like extremophiles anyway, so they're like molecules that exist in really extreme conditions, not necessarily all of them, and in fact we don't really know. These things the archaea is a much later construct, as in discovered, than bacteria was, so yeah. So the next sort of thing that we're going to talk about is, so these are domains, important to remember. Okay, so we've talked about classification. Now let's talk about structure. Structure is very important. Structure and composition. Try and make this as nice as possible. Structure and composition. So, how are prokaryotes structured? Terrible box, sorry about that. So, how are they structured? It's basically like, imagine it's a cell that has, it's like it has a membrane. So okay, so here I'll show you. Let's focus on. Let's go here. Cell membrane, as I mentioned earlier, bilayer of phospholipids. Right. There is a circular DNA molecule. So this DNA molecule is basically like, this is the DNA of the thing. It's circular. It's fairly fairly simple DNA. There's a zone called a nucleoid. So this is a high concentration of DNA. Like most of the DNA exists, I think most, if not all, of the DNA exists in the nucleoid. What else is there? We talk about ribosomes. So there's a thing called a cytoplasm. So this is like it's got raw materials and stuff is floating around. Molecules floating, right? The function, I mean, the cytoplasm just holds everything. It's like a jelly, basically. Now, in the cytoplasm, there are also, this is important to the thing, ribosomes. So they synthesize. What do they synthesize? They synthesize proteins. Very important. Very, very important. Now, there's also like some other functions that are pretty there's certain structures that are more or less unique to prokaryotes. Unique structures. Yeah. Sorry. So they have a cell wall of peptidoglycan. So this is a rigid one. And they have a cell capsule. Which is like polysaccharides. It's like sticky for cushioning. It like sticky and it like cushions the cell. That's cushions. So the cell wall, it's rigid. The cell capsule, is like spongy, I guess, sticky. Now, there's also, this is not unique to prokaryotes, but there is something called a flagellum. So a flagellum is so it comes from the Latin for whip, and it basically it's like a proteinous structure that allows from the membrane that allows movement, or it's motility, that's how it's called, motility. Yeah. Yeah, so that flagellum also exists. It's a proteinous structure. It's not unique to prokaryotes, but some prokaryotes have them, and they have multiple sometimes too, so just remember that. So unique structure to prokaryotes are the pili. They're called fimbriae. I think that's how you spell that. And they're like hair-like projections. Hair-like projections and they're for adhesion. For adhesion, yeah. All right. So these ribosomes, let's talk about them for a second. They synthesize in two places. So it's either, in the cytoplasm or cytosol, the jelly is called cytosol, or in eukaryotes on the endoplasmic reticulum. I'll talk about that later. So this pillar, that's a unique structure. Of course, it's very important to note, and this is something that should come up a few times, is just how much more complex. So the complexity of a eukaryote is just much higher than a prokaryote. A eukaryote has, like these massive, eukaryote is much more complex than a prokaryote. Yep, it's just very important. So like, for example, a ribosome. Eukaryote, it has about 88 proteins, or 80 proteins, really. Synthesizes those. And then the ribosome of prokaryote cannot do as much. It's about 55 proteins. The point here is just that the complexity is much higher, the prokaryote. Okay, let's talk about the function and abundance. All right. So function and abundance. So what do eukaryotes do? And what do prokaryotes do? And how do we sort of use them as well as humans? Because we're the, the apex predator, I suppose. But so when we talk about prokaryotes, so let's talk about abundance first because it's relatively smaller. Basically, the point here is prokaryotes are everywhere. There are some at like really extreme really extreme conditions. So really high temperatures and acidities and very low acidities. They still exist, like, you know, these archaea, for example. Some archaeans are really you know, exist, you know, 100 degrees, for example. We talk about, there's a very good homeostasis. That's the point. They can live anywhere because they've adapted to their environments. They've evolved over time so that they can just live without any problem, you know, at the bottom of the ocean or wherever they want to, you know. Even the stuff in your gut, the gut microbes, the bacteria there has evolved to live in your gut and helps you, because if you're a better organism, then they live better. Yeah. There's some like vague thoughts as well about other types of prokaryotes, because they're so abundant. It's potential, like maybe you could have a prokaryote that doesn't use doesn't use oxygen or anything, or even that generates methane for its waste instead of producing instead of using water. Water and carbon dioxide uses hydrogen, carbon dioxide uses methane and produces some energy. That's just a thought, really, and I'm obligated to put it in here. Thanogenic bacteria. Yeah, cool idea though. Okay, so let's talk about function now. This will be a bit longer, but still, last topic, very good stuff. I think I'm running on good time. So function. Here's a little side point. They live on us. Look at the little cheeky buggers. Everywhere. Absolutely everywhere. Tips of your fingers everywhere. You can never escape bacteria. Why would you want to, though? They're so nice. Okay, function. So when we talk about function, there's like bacteria, like they perform different. They, like, for example, they can like eat toxins. So they can eat toxins. We can genetically modify them. To produce better crops. To produce pharmaceuticals. Pharmaceuticals. What a word. Pharmaceuticals. You know, there's like we can have genetic modification for crops. So you can have, like, we use them all the time as humans for different things. Like, if they eat toxins, right, we can use them in, you guessed it, waste treatment. If we use them to produce pharmaceuticals, like insulin. Insulin was one of them. So basically this process is prokaryote DNA switched, like changed, for whatever codes for the, like, the desired substance. Prokaryote this is pretty cool stuff, to be honest. This pharmaceutical stuff, like we can produce insulin to help with diabetes, or we can grow GM crops that are pesticide resistant because they've had exposure to the bacteria. And yeah, we basically just insert the desired genes we want into prokaryotes, and then they produce what we want. Toxins as well. This one is bacteria in general. I mean, they eat toxins because they just work through the waste. Yeah, so that's just the sort of functions. You can have waste treatment. We can have sort of genetically modified stuff, and that's the sort of the functions of prokaryotes that we can do. But I mean, the prokaryote itself, it functions. It's a cell, right? So it's cell's function is just to reproduce, really. We can do all these things because we are very smart humans. And they're everywhere. They're absolutely everywhere. I just know they've got good homeostasis. They got to be, like, very well optimized creatures, in organisms, in order to be able to live in these all these places, and they change. So basically, yeah, we talked about classification, structure, composition, and function and abundance. I think that's everything. Just remember a little summary: no nucleus, no organelles, pretty much all unicellular, and they're like the first cells. Basically, they're like the simplest form of life that we know now, like life, like very, very, very simple cells, but ultimately ones that also have evolved over a very long time, and have differences and unique structures and tons of different things which make them what they are. Okay, cool. See you in the next one.